Hi everybody, and also welcome for me. So my name, as you already heard, is Sebastian Bludau. I'm a member of the team of Katrin Amons in Jülich, and I would like to talk about brain atlases. So at the very beginning, I would simply clarify the wording. So brain atlas, of course, something comparable to a normal map. So we have the Earth as one coordinate space. Of course, in our case, it's the brain, the coordinate space we are working with. And we do have different parcellation schemes for those coordinate spaces. So for the brain, as Katrin already presented, for example, it could be the Jülich brain atlas, which is built on cytoarchitecture, comparable to a classical political map you can see in the upper row there. All those areas or countries can be grouped by a predefined taxonomy. For example, for the brain, if you group them via brain lobes or something like this, you can do the same for the political map with countries and continents. So it's highly comparable. And like for the brain, we also have different types of data which could be integrated into an atlas. So Katrin already raised the point with the Google Maps example. So there's the political map information, there's topographic information, climate information. All those information can be added into an atlas. And yeah, you have seen this slide before, so Katrin already spoiled it. So we have different reference spaces which are already integrated into the EBRAINS infrastructure. So they are all in the same coordinate space, but they are different reference spaces. Here, for example, we see the parcellation of the Jülich brain atlas, and we can directly switch in the browser, and in the viewer we'll see it later on, to switch to the just presented fiber bundle atlas or the functional modes, which are also already included. It's a bit more complicated for brain atlas, so we do not only have one planet Earth, we have different planets, so we have different species here. So we have the human brain atlas, but also the rat and the mouse atlas and the monkey atlas, which will come in the near future. So just as a summarize of the very beginning that we have the same wording for the talk. So if you talk about an atlas, it's something comparable to the species or to the planet, if you will still stay in the example with the map. So I will focus on the human brain atlas, and this is the topic we are working with. Then we have different parcellation schemes, like the Jülich brain atlas or the Desi-Canckelliani, the FUMO. This is comparable to countries, continents, or even a time zone parcellation of the Earth. Then we have different reference spaces. This can be compared to different coordinate systems. They describe the same planet, but you can either take a map or a globe. It depends what you like, what you prefer, but they describe the same coordinate space. We mostly use those standard reference spaces, for example, the MNI 152, which is one commonly used reference space, or the FS average space, which is the surface-based representation of the data. It's integrated in the free surfer pipeline. And we also have, as already mentioned, the big brain high resolution reference space. And finally, there are those regional features which enrich the data of the areas or of the countries. So like cell densities, receptor densities, functional data. It's comparable to the number of inhabitants, something like this. So additional data. If we want to work with the atlas, so you can easily find the eBRAINS webpage via Google or whatever you prefer. Then you have to search for services, and there will directly find the atlases. In order to make the data available, there are three major points which have to be fulfilled from our side. So we need software interfaces to access the data. Those are the atlas viewers and Python library we've just heard. We need tools to integrate the data, to make it available. And of course, we do not only want to see or visualize the data, we want to work with the data and really include it in our experiments. And therefore, we need tools to analyze the data. So yeah, let's start with the software for accessing the data. This is the interactive atlas viewer. The address in the middle, you can directly go through it. It's completely free, as we've heard. Here we see the Jülich brain parcellation in the MNI 152 reference space. So all three two-dimensional panels are connected. So it's a 3D data set. On the lower right side is a 3D representation of the map. Now we have switched to the MNI colon 27 reference space. It's also a commonly used reference space. And You can see sometimes the areas are highlighted as if you hover with your mouse above it, you can receive further information which will be shown later. The third reference space which is available is the FS average representation. The difference here is that it's a surface-based representation. So the first ones were volume-based representation, and this is surface-based. It depends on your need. Sometimes the surface-based parcellations are easier to handle, and one benefit that you can inflate. So, for example, if you want to visualize areas located down in the side, you can easily use this inflated version. Of course, the benefit from the volume-based representation is that you can also analyze and visualize subcortical structures. At the end, it depends on you, on your need, what you prefer. So we have already heard about the different scalings. And yeah, we would like to bridge between the microscopic and the macroscopic scale. And one thing which is already implemented in the EBRAINS infrastructure is the linking between the high-resolution big brain model and the classical reference spaces like MNI, colon or 152. So the big brain model is a model which is based on 7,400 histological sections. All of those sections were digitized with 20 micrometer in-plane resolution and finally reconstructed. So by doing this, we received a model, a dense model of one human brain with an isotropic resolution of 20 micrometer, which means we have 20 micrometer in all three directions. And this high resolution model is directly linked to the classical reference spaces, so you can interactively switch between the reference spaces and see the microstructure of a specific area coming from the classical reference spaces. It also depends on your need. If you need a higher reference space to anchor your data or the lower resolution is enough, yeah, it depends. So that we can switch and that we really can see high resolution data, you can see by this example here. So if we search, for example, for the primary motor cortex, it's called area 4A in the Jülich brain atlas. You can see it's highlighted light blue here. And up to now, you have to believe me, it's the motor cortex. But if we switch to the higher resolution of the big brain data set, we will have the benefit that we have 20 micrometer in-plane resolution, and we can zoom in. And one of the very specific cytoarchitectonic features of the primary motor cortex are the so-called Betz giant cells, which are only visible in the primary motor cortex. And the 20 micrometer resolution are enough to see those Betz giant cells. So the red cursor highlights here the giant cells normally located in lamina 5 of the primary motor cortex. Of course, this is a very easy example. We do not have such prominent histological features in every area, but here we can directly benefit from the higher resolution. Okay, so the second point I raised at the beginning was the integration of the data. Of course, it's very important to bring the data in a structured way. And those additional features could be, for example, high resolution connectivity data like the PLI data, which is located here. We have high resolution cell densities anchored in different areas. We have receptor densities, but there's also functional data available. So, but all these kinds of data have to be integrated into the atlas. And one starting point would be, for example, that you start anchoring your slice sections or incoming volumes in an interactive way. This can be done, for example, for the mouse brain using the QuickNey tools, where you interactively anchor 2D sections in a reference space. And by bringing your data into this well-described reference space, you can directly benefit from the available information there. Another tool which is already available is the Voluba tool. It's comparable, but in 3D. So with this tool, you can interactively anchor incoming 3D volumes into the big brain reference space. And by doing this, so the data sets become real curated data sets. So Katrin already mentioned the FAIR data set, which means it's findability, accessibility, interoperability, and reuse. So that the data set is not only a label placed somewhere, so you have really precise meta information behind the data set. You have embedded information which you can directly download and use and work with the data. I think the movie on the right side we have already seen. So in order to provide such data sets, of course, we need a curation team. Katrin already mentioned that we will hear more about the curation of the data sets. So here, very short, there's a curation team which helps you to integrate and bring your data into the EBRAINS infrastructure, because it has to be structured somehow. So as you said, we want really deeply curated data set. We do not only want to store it somewhere and give it to the people should be able to work with the data, and therefore they need a lot of information, because it's really difficult to work with scientific data which was not created by yourself. And therefore all this information we try to integrate and make it usable for the people. So then we have this knowledge graph kind of database where you can search for your data and browse it and search for specific keywords. And this knowledge graph is directly interacting with the Atlas services. So the Atlas viewer we have already seen, and also the Python library I will present you later. So they directly interact with the EBRAINS knowledge graph and get all the data and information and metadata from this knowledge graph, which makes everything work as a complete workflow. So once everything is there, we really have such deeply curated and linked data sets. So on the right side, you can see they have selected a specific area. They can browse for regional features, select those features, get further information. They will, if you click on one of the buttons over there, you can directly go back to the knowledge graph and get there further information. You can download it and really work with the data. And this is something we'll also do in the hands-on session later. So all the examples I'm showing here will be done later. They are really easy, and everybody would like to invite to join this hands-on session, because it's really on the beginner level, and we would like to introduce the tools and the workflows. The last point is, as I said before, we do not only want to visualize it, we want to work with the data. And therefore, we need different ways of accessing the data. One way, the very easy way, is the Explorer, the siibra Explorer. We have seen it a lot. The other way, which gives you more degrees of freedom, is the siibra Python library. It is very easy, but of course you need a bit of program coding to work with it, but it's not that complicated. And the siibra Python library really gives you full access to all the data which is created in the knowledge graph. And you can build your complete workflows, you can download the data, work with the data, build complete experiments, and enrich your own data with the data which is already there. And for those who are interested in this, we have several examples. As Katrin already mentioned, we can try to set up some of those scripts. We can directly use them in the browser. So it's really easy for everyone, we hope. One of the examples is a UJAX tool. We have heard about it. So it's a quite deeply curated tool in the Atlas viewer and in the Python library as well. So it uses the Jülich cytoarchitectonic maps and extracts additional genetic information, which is also free available via the Allen Brain Institute. So they provided about 3,600 tissue samples, which were fully analyzed for the gene expression profiles. And we could use the maps integrated in the Atlas as a kind of mask to extract only those tissue samples which belong to a specific region. And we can directly analyze them for differential gene expressions in the browser. And this can also be done via the Python library. So there's really very easy tools. It's a plugin. You can click on an area and integrate it into the experiment. You can freely define the genes or the gene set you would like to analyze and directly perform the analysis in the browser. Although the browser plugin is the basic version, so you cannot do full experiment in the browser because it's too compute intense. But if you really want to create an experiment you could use for publication, you should switch to the Python version, where you have every parameter you could change for the number of permutations or a longer list of genes, whatever you need. So there you have the full degree of freedom. So yeah, coming to an end, I would like to summarize some of the points you have just heard. So the data sets are fair data sets, or really they are deeply curated. You have a lot of information, you have an easy access to the data, and you can use them for reproducible experiments. There are different interfaces to work with the data. The easy starting point using the browser version in the Zebra Explorer, or the programmatic way using the Python library, depending on your needs. And by integrating the data itself and the atlas in your workflow and in the large environment, you can directly benefit from the large amount of information which is already available in the EBRAINS infrastructure. Yeah, and I think it's a win-win situation for everybody. So if you bring your data into the EBRAINS infrastructure, you can benefit from the information which is already there. And of course, the other people can benefit from your information. So it's really the idea of making it openly available and work together. So then I would like to thank you for your attention. And again, I would like to also invite you to our hands-on session, where we can do all the stuff together and browse through the atlas, whatever you would like.
Yeah.
Thank you.
Thanks so much.